Welcome everyone to this week's lecture. In previous lectures, we've covered T cells at length from antigen presentation, activation to the effector mechanisms of T lymphocytes or cell-mediated adaptive immunity in general. Now we're going to cover the humoral immunity arm. So first we're going to start in this week, we're going to talk about B cell activation and antibody production. Next week, we're going to talk about the effector mechanisms of the humoral arm in general. And I'm getting excited because after that we can start talking about ways in which the body is deficient in these mechanisms and diseases and the way engineers used to treat it. We're going to talk about cancer, autoimmune diseases, and a whole host of other ways where we can bring up, again, the engineering arm of biocompatibility. Now, we've covered some of this in previous chapters, so I want to give a brief recap and, I guess, general overview of what this entire chapter covers. So first, we know that mature, naive B cells that are in circulation don't secrete any antibodies. They only have membrane-bound antibodies, which act as receptors. And these are typically IgM or IgD antibodies. So you wouldn't see a naive, mature B cell, for instance, with a IgG surface-bound antibody as a receptor, only of the M and D varieties. We also know that upon activation, a single B cell can reproduce and expand to as many as 5,000 cells. We learned that this is in contrast to T cells, which can expand up to 50,000 cells. And also that these individual antibody-producing B cells after activation can produce, 10 to the 12 antibodies per day, an insane amount. They're just huge factories for antibodies. When covering the T cell effector mechanisms, we learned about follicular T cells and that B cell activation can sometimes require T cell help. So in this case, activation can be divided up into T-dependent activation or T-independent activation. So the T-dependent are the ones that require help, aptly named, from T helper cells, specifically the follicular helper T cells. Just like how naive B cells typically only express IgM or IgD subvariety of membrane antibodies, upon activation, they'll typically only secrete those same antibody subtypes, IgM and IgD. We also learned of the possibility of undergoing what's called isotype switching, where the B cells are told to change which type of heavy chain or the class or subtype of antibodies to produce. And this is how they can start producing things like IgG, where they weren't before. We also talked about the process of affinity maturation, in which the genes that encode the membrane-bound and secreted antibodies are mutated ever so slightly so that they still retain the same general affinity towards their antigen, meaning it doesn't change what antigen it's specific to, but the affinity towards that particular antigen is drastically increased, up to 2 orders of magnitude. And then lastly, we also understand the process that in producing effector B cells, meaning ones that produce antibodies, it's also possible to produce memory B cells in the same way that T cells upon activation can produce effector T cells of the CD4 or CD8 plus variety or memory T cells. Now here we have a brief overview of T-independent B cell activation. So first of all, what's required for T-independent activation are antigens with long repeating epitopes that are capable of binding multiple binding sites on a B cell. So let's stop imagining that just one receptor binds to one antigen and that activates the B cell. Now I want you thinking, okay, we have a really large molecule like a polysaccharide, a polypeptide that has these repeating epitopes that are all exactly the same, which means that they can bind to the same receptors, the membrane-bound antibodies on the B cells. And that's what's required for T-independent. For T-dependent, it requires a lot less help because the T cells are very good at their job of helping the B cells. So, long repeating epitopes, like a polysaccharide, a polypeptide, can activate a B cell. This is a rapid, but it's a relatively simple process, in part because the secreted antibody, which is usually IgM and to a lesser extent IgD, has the same exact affinity as the original membrane-bound antibodies. So there's no affinity maturation here. There's no class switching. Furthermore, there's no production of memory cells. We'll talk about the need of T cells in order to produce memory B cells. And this is primarily carried about by 2 B cell subsets. So marginal zone B cells, which are in the spleen, and then B1 cells, which are mainly mucosal sites. In contrast, we have the much more exciting, much more sophisticated T-dependent process. And I actually learned that in this case, helper T cells were called so helper because scientists originally found out that their need to help B cells to do all this cool stuff. So even though we learned about the helper T cell roles in macrophages, neutrophils, eosinophils, and whatnot, it was here where they originally got the name helper. So anyways, so T-dependent is required for all protein antigens. So yes, B cells can recognize peptides and proteins, but they need helper T cells' help in order to process those protein antigens and get activated. T-dependent activation is required for class switching, so in order to change the IgMs to IgGs, IgAs, IgEs, and so on. This also stimulates the production of long-lived plasma cells and memory B cells, so you need T cells in order to have memory B cells. And this is primarily carried out by the subset of B cells called follicular B cells, which we'll learn are in the follicle zones of the lymph nodes. So next, let's talk about how antigens are captured and then delivered to B cells for activation. First, let me remind everyone that B cells can recognize all types of antigens, right? Whether they're large, small, particulates, solubilized, lipids, carbohydrates, proteins. This is in big contrast to T cells, which require cell-presented only peptide antigens that are attached to MHCs. So this is a big difference between T cells and B cells. Anyway, so most antigens are transported to the lymph nodes by afferent lymphatic vesicles. Okay, soluble antigens that are less than 70 kilodaltons can reach the B cell zones through just normal conduits. Larger antigens and antigen-antibody complexes sometimes have to be delivered to the follicles via subcapsular sinus macrophages. So these are a specialized macrophage subtype that just live in these lymphatic organs. Large soluble antigens may also be transported to follicles via specialized dendritic cell subsets in the medulla. And there's also a few other things. So antigen-antibody complexes can also be carried by marginal zone B cells or follicular dendritic cells via this complement type 2 receptor, CR2. And then polysaccharide antigens can also be captured by macrophages in the spleen, lymphoid follicles. Now, in all of these cases, what I want to point out that as opposed to T cell, where there's a specialized antigen processing mechanisms that chews up the proteins into small peptides and then onto MHC complexes, for B cells, all antigens are presented in their intact native form. So there's no mechanism to trim them, snip them up in specialized ways for the B cells. They always take intact antigens. Now, the actual activation of B cell has what I think are a lot of similarities to T cell activation, which I'll describe in just a sec. But first of all, the B cell receptor complex isn't actually composed of singularly just the membrane-bound antibodies. The membrane-bound antibody, which has the two binding sites, and then these Ig alpha and Ig beta proteins. Now, beyond this, I'm not going to talk about this complex in any more detail with the alpha and beta chains. Suffice it to say that they're required for some of the intracellular signaling processes, but I'll just leave it at that. Anyways, the binding of this receptor to its antigen serves two purposes. The first is that it initiates signaling for activation, and the second is that this complex will then process and internalize that antigen through endosomal vesicles, and if the antigen is a peptide or a protein, then it's going to digest it and present it on MH2 complexes, just like other antigen-presenting cells. Now, when I mentioned earlier about the similarities between T cell and B cell activation, I was thinking of the signal 1 and signal 2 steps for T cells, in which you need both the antigen recognition and then a co-stimulator. Now, the book doesn't use that terminology here with B cells, but something like that absolutely is necessary. In fact, they say co-receptor here, but B cell activation is facilitated by the attachment of these co-receptors. Like CR2 is a very common one on B cell. It's a complement receptor that recognizes a specific complement factor, C3d. You don't necessarily need to recognize or memorize that right now. We're going to talk about complement much more in the next chapter, but it's just one of the byproducts of the complement process, which I described a few lectures ago. So just like the picture shows here, we have the antigen being recognized by the B cell receptor, and then we have the CR2 co-receptor recognizing the complement protein. And that all enhances the B cell receptor signaling process and initiates activation. Other pattern recognition receptors, like toll-like receptors, are present on B cells that aid in proliferation, differentiation, and activation of B cells. B cells have a more limited variety of toll-like receptors than other cells like dendritic cells, but they do have abundant amounts of toll-like receptor 5 for bacterial flagellin, endosomal toll-like receptors 7 and 9, which recognize nucleic acids. And then they can also indirectly be induced to activate potentially by RA-activated dendritic cells, which can help better activate helper T cells to promote the activation of B cells, or other myeloid cells that, through their own toll-like receptors or pattern recognition receptors, can secrete cytokines that can aid in the activation process. As I mentioned earlier, it can help if a lot and abundant clusters of the B cell receptors can also bind simultaneously, like is the case with any antigens that have large repeating epitopes. So think of polysaccharides that can cross-link multiple B cell receptors simultaneously. Now note that this doesn't necessarily happen with large proteins that don't have repeating epitopes. So they might be made up of long chains of amino acids, but unless those amino acid clusters that can make up an epitope are repeating, then that isn't going to happen with proteins. However, this doesn't prevent B cells from simply recognizing large proteins. There's some evidence that this might stimulate them to continue surviving, perhaps when an innate immune response might help enhance the activation process. And also, this doesn't prevent the B cells from internalizing them and then presenting them, hopefully, for a T cell as well. So remember, they can act as antigen-presenting cells as well. So that requires a lot of the necessary steps for T-independent activation. So now let's get into the T-dependent activation processes. And first I'm going to cut it to a video from the book, and then we're going to talk about it a little bit more in detail. Enjoy.
The process of T-B cell interaction and T-cell dependent antibody responses occurs in a series of sequential steps. First, CD4 helper T cells and B cells are independently activated by a protein antigen in different regions of a lymphoid organ and migrate towards each other. Second, this initial migration or interaction between T cells and B cells takes place outside the follicles. Third, the initial antigen-specific T-B cell interaction consists of two phases. A, B cells process and present antigen to the T cells. And B, the previously activated helper T cells express CD40 ligand and secrete cytokines, which act on the B cells to initiate proliferation and differentiation to plasma cells. The early antibody response, consisting of antibody secretion by plasma cells and some degree of isotype switching, occurs in these extrafollicular foci. Fourth, some activated B cells migrate back into the follicle, accompanied by helper T cells that were further activated by B lymphocytes to develop into follicular helper T cells, known as TFH cells. In response to signals from the TFH cells, B cells begin to proliferate, forming an organized structure called a germinal center, in which stronger and more effective antibody responses develop.
Now, as the video just stated, in the process of T-dependent B cell activation, both the T cell and a B cell get independently activated initially, and then they have to meet up. First, let me clarify that the epitope that activates those two cells independently are not the same. Okay, it might be possible that's the same antigen and there's different epitopes on them that activate the two cells separately. It might even be possible that it's two completely separate antigens. And we'll talk about that in a little bit of detail in a few slides, but I just want to point that out right now. I'd also like to take just a few seconds and appreciate the sheer amount of coordination that's involved in getting both of these independently activated cells to meet up with each other in the relatively large lymphatic organ here. Not only is it a large distance to cover, but there's so few cells that are specific to any one epitope or antigen. It could be 1 in 100,000, 1 in 1 million. And they still have to meet up with each other. So I like to think of it as a person in Los Angeles being told to meet up with a person in New York, somewhere in the middle of the United States. So let's discover how these cells actually do that. So T cell activation, we already kind of know, right? Dendritic cells, they process antigens via endosomes. They digest the proteins and display the peptides on class 2 MHCs. This activates the CD4 plus cells. Activation causes a downregulation in the chemokine CCR7. And because I do it every time, I feel like I can't go on without saying my little memorization technique of Creedence Clearwater Revival or Cristiano Ronaldo, who wears the number 7 jersey. But anyway, CCR7, incredibly important chemokine. It recognizes the ligands CCL19 and 21 that are expressed in the lymph nodes. That's what helps dendritic cells and T cells get within the lymph nodes and meet up at the same place. So activation downregulates that because now they no longer need to be in that T cell zone within the lymph node. In this case, the T cells, after they downregulate CCR7, start increasing the expression of the chemokine receptor CXCR5, which recognizes the ligand CXCL13 that's expressed by follicular dendritic cells that are in the follicle. If you have trouble remembering all these ligands and receptor numbers, I just remember lots of odd numbers, lots of prime numbers, and then just try to remember the pairings for each one of them. So the net result in all of this is that the T cells leave the T cell zone and head towards the follicle where there's a higher concentration gradient of the CXCL13 ligands being expressed by these dendritic cells. Now, if you got that, the B cell stage is actually pretty easy to remember because I think a lot of it is just the reverse process. So in this case, B cells, they bind to a protein, and that initial coupling initiates the start of activation. The B cells then process that protein and then present the shorter peptide sequences on class 2 MHCs. Upon activation, B cells reduce their already CXCR5 chemokine receptor and then they start expressing the chemokine receptor CCR7, which again binds to CCL19 and 21 in the T cell zones. So this is the exact opposite of T cell expression. So then the net result of this is that B cells want to leave the follicle and they head towards the T cell zone. So in short, they're both going opposite directions towards each other. Because of these opposing migratory patterns, eventually the T cell meets up with the B cell and then activation continues. So I mentioned before that the epitopes that activate B cells and the T cells, the CD4 plus cells, are not the same. So let me illustrate and expand on that concept a little bit more. So the surface conformational epitope is what the B cell recognizes. Remember, it can recognize solubilized proteins, different conformations, whatever is in the natural state of the protein, that's what it recognizes. It doesn't have to recognize short little peptide sequences. So that's what starts the activation process in B cells. Part of that activation process also means that the B cell internalizes it, processes it via endosomes, and that's why that particular peptide ends up on class 2 MHCs. Now, on the other side for the T cells, dendritic cells would have already done this. They would have broken down the same peptides, the same proteins, made the peptide shades on class 2 MHCs, and activated T cells independently. Now, the B cells, just like other antigen-presenting cells, don't know which short little peptide sequence is most important for T cells. So they just break it up and they express class 2 MHCs with all sorts of the little peptide chains that make up the larger protein that originally activated them. So that way they have a better chance of presenting whatever magical peptide sequence that is to the proteins, and while simultaneously expressing all other short little peptide sequences that are made up of that larger protein. So in this particular scenario, the same overall protein activated both B and T cells, even though the epitope was different for the two respective cells. This also brings up a second scenario in which the B cell and T cell activation does not occur on the same antigen. And engineers can make use of this through what's known as the hapten carrier effect. So haptens are small, non-immunogenic molecules that bind to B cells but do not in themselves induce strong B cell activation, in part because they're not coupled with a strong innate immune response. They don't have pathogen-associated molecular patterns, anything to help otherwise initiate B cell activation. However, if you conjugate a hapten to a protein, so you make a hapten protein conjugate, those are capable of producing an immunogenic effect because the T cells recognize this and aid in B cell activation. So the hapten carrier effect requires three things. One is that there's a hapten that's specific to B cells. And remember, they could be non-immunogenic just as long as the B cell surface antibody recognizes it. Second, the hapten has to be conjugated, physically linked to the protein. These can't be administered separately. Third, the T cells have to recognize a peptide sequence on the protein. So this is the carrier epitope. If all three requirements are met, Then the B cell will present that particular peptide sequence to the T cell that will cause activation of the T cell and then help activate the B cell further, and then the B cell will produce antibodies specific to the original hapten. If it was just the hapten, the B cell might still recognize it because it has surface antibodies that can recognize it, but it would not be induced to activate and then turn into antibody secreting B cells specific to that particular hapten. And again, scientists and engineers make use of this to develop conjugate vaccines that will produce strong antibodies for non-peptide sequences because they made use of that sort of protein-specific, the peptide-specific recognition of T cells to help augment the antibody production of B cells. So before I introduce the next step of the B cell T cell activation process, I'm going to bring us back to week 5 when we were talking about T cell activation and the role of CD40 and CD40L in that process. So if you remember, the T cell here, the red cell, has the CD40 ligand, and that's what attaches to the CD40 receptor on the dendritic cell. And here the slide even says that leads to quote-unquote D cell activation. Now what that means is that upon the dendritic cell recognition via its receptor of the CD40 ligand, the CD40 receptor initiates intracellular signaling processes that causes for the increased expression of the B7 ligand, which can then help promote T cell activation and proliferation and differentiation. Now, the role of CD40, CD40L comes full circle into B cell activation. So in this case, think of the B cell as taking the place of the dendritic cell in the previous slide, which is very appropriate because B cells themselves are antigen-presenting cells. So here, the T cell expresses the CD40 ligand, the B cell expresses the CD40 receptor. Now, B cell proliferation and differentiation is caused by two things: the direct binding of CD40 via CD40 ligand on the T cell, and the indirect secretion of cytokines by the T cell that it in itself is induced by the binding of the T cell receptor MHC antigen complex. And that causes the T cell secrete these cytokines, which further enhances the activation proliferation process in B cells. Now, all this is happening at the interface between the follicle and the T cell zone. Now, all subsequent activation, so we're still in the activation process here. It's like a very many-step process. Subsequent activation between these two cells occurs at two different locations. One outside the follicles in the extrafollicular focus, and then the other in the germinal centers of the follicles, which is where all the cool stuff happens. Now, I like to think of the extrafollicular B cell activation as just about as boring as this slide. There's nothing really going on with this slide. It's kind of bland. There's no pictures. And I think it's the same way for the reactions that are in the extrafollicular zone, despite them being involved with T-dependent reactions. So in part, it's important for the early response of these type of B cells to protein antigens. They generate antibody-secreting subtypes of B cells that are really short-lived, so they don't last long. These cells are called plasmablasts, which are really just immature plasma cells. Plasma cells are the effector versions of B cells that produce antibodies. They generate low-affinity antibodies that circulate and help limit the initial spread of an infection. And there can be even some minor class switching at this stage, although it's very minor. And then we get to the T-dependent B cell activation in the germinal centers, which is a lot more exciting, hence the exclamation mark, yeah. So in short, this is where all the cool stuff happens that are associated with T cell-dependent antibody responses. Okay, affinity maturation, isotope class switching, the generation of long-lived plasma and memory cells, it all happens here. There are two areas of germinal centers called the dark zone and the light zone. The dark zone is filled up of just B cells, and this is the site of rapid proliferation of the B cells. The light zone has B cells in addition to two characteristic cells of germinal centers. One is the follicular helper T cell, and this is what the helper cells differentiate into once they're inside the germinal centers. And then there's also follicular dendritic cells. So follicular dendritic cells are unique to dendritic cells because they lack class II MHCs. In order to compensate for this and still display antigens to the B cells, they express complement receptors 1 through 3, and then also FC receptors that help them display antigens. So remember, despite their name, these dendritic cells don't share very many similarities with the dendritic cells that we've talked about all throughout class. In fact, they're not even derived from hematopoietic stem cells. in the bone marrow. So they come from a different cell lineage. Now, within the dark zones of the germinal centers, this is where B cells undergo what's called somatic hypermutation, and this is the mechanism that drives affinity maturation, which, as the name implies, results in higher affinity antibodies to these B cells. Now, the entire formation of these germinal centers with high concentration of B cells is dependent on the CD40 ligand of the follicular helper T cells interacting with the CD40 receptor on B cell. Not only this is critical for B cell proliferation, but also induces the expression of an enzyme called activation-induced deaminase, AID, which is required for both isotope switching and affinity maturation, which we'll talk about in a little bit later. Now, the general process between migration from the dark zone to the light zone and back is that dark zone is where proliferation happens. Then the B cells go into the light zone. They interact with the follicular helper T cells. B cells with higher affinity antibodies go back to the dark zone to proliferate, and then the process is repeated multiple times. Afterwards, those B cells selected to continue are turned into plasma cells, which are the effector B cells that produce the antibodies, and then also memory B cells, and they both exit the germinal center. Now, because one of the characteristic cells of these germinal centers are the follicular helper T cells, I want to talk about them and their differentiation a little bit more in detail. So, prior to activation, these T cells are just CD4 naive cells. In order to turn into these TFH cells, they have to first get initially activated by dendritic cells in the T cell zones, and then undergo subsequent further activation by B cells. On the T cell side, differentiation to TFH can be promoted as opposed to the Th1, 2, or 17 subtypes, in part by a high peptide to class 2 MHC T cell receptor binding strength. Once again, to help ingrain this into your memory, at this point the T cells start expressing higher concentrations of CXCR5. That's again in part promoted by the high peptide class 2 MHC T cell receptor binding. And this wouldn't necessarily happen with other T cells that had lower strength binding and then would become Th1, 2, or 17 subtypes. So anyways, they start expressing the CXCR5 receptor that attracts them to the CXCL13 expressed by the lymphoid follicles. B cells, on their side, once they interact with these T cells, they can help promote the differentiation of the T cells into the follicular helper T cells through the binding of the adhesion molecules ICOS-L on the B cell side. So again, that's the ligand. So the B cell doesn't care that that's binding, but the T cell that has the receptor for ICOS-L, just the ICOS receptor, does care. And so whenever that binds to its ligand, it initiates downstream signaling processes within the T cell zone, and it promotes the differentiation into the follicular helper T cell subtype. Now, just like we know the signature cytokines of the other three subtypes of T cells, 1, 2, and 17, is IFN gamma, IL-4, and IL-17, the signature cytokine for this helper T cell is IL-21. This is an essential cytokine for germinal center development and also contributes to the generation of plasma cells. So now let's talk about class switching in detail. So antibody class switching occurs in the germinal centers, specifically in the light zone areas, because it's driven by the follicular helper T cells. The purpose of class switching is to adapt the B cells to express whatever antibodies that are required for an appropriate infectious agent or an appropriate function. And that function is always guided by whatever infectious agents are present. They somehow induce through innate immune reactions or other mechanisms to instruct the helper T cells to then instruct the B cells, all through cytokines, to tell them what antibodies to switch to, right? So for instance, helminths could induce the follicular helper T cells to produce cytokines that switch the B cells to IgE antibodies, which we learned are required to fight against IgEs through eosinophils. Or another instance, B cells could be instructed to produce IgA antibodies, which are important for mucosal immunity. So whatever antibodies are necessary to fight off that particular infection usually gets word through the helper T cell to then get word to the B cells to class switch to that particular antibody producing type. And again, the two things that mediate this are, first of all, the cytokines that give the signals what to switch to, and then in order to switch in general, the CD40, CD40L binding, which, as I mentioned earlier, introduces the expression of the AID enzyme, which is necessary for this process. The other really cool process that happens in the germinal centers is affinity maturation. So again, previously, especially like in the extrafollicular T-dependent reactions or the T-independent B-cell activation processes, B-cells produce just really bland antibodies that have the same exact affinity as the original naive membrane-bound antibodies. But in this case, through affinity maturation, the B-cell antibodies could potentially have stronger affinities up to 10 or 100-fold times stronger than the original naive B-cell. This makes them much more effective at carrying out their jobs. So let's talk about it a little bit more. In general, it's the process of having an increase in affinity of the antibodies for that same particular antigen. And it's really the result of what's called somatic mutation of the variable genes of the antibody. So these are the parts that bind to the antigen. And then the selective survival of the B cells that produce antibodies with the higher affinities as opposed to lower ones that result from this mutation. Now, at this point, if I were to ask you, hey, if you had a guess, what's required in order for this to happen? I just want you to start guessing CD40, CD40L, because it's involved in everything, and it is indeed involved in this case. Now, because the CD40L ligand is required, that's another clue that T cells are required, and that affinity maturation can only happen in cases where you have a protein antigen, because otherwise, without proteins, T cells wouldn't be involved. The mutations occur during the proliferation and reproduction process, so because of that occurs in the dark zone, not the light zone of the germinal centers. And it's really just single point mutations that rearranges the Ig variable genes that encode the variable region of the antibodies. And this occurs at an extremely high rate. So to give you an example is that point mutations in just normal mammalian genes usually occurs at a rate of around 1 in 1 million gene base pairs per cell division. Except here, it's occurring at a rate about 1 in 1,000. So that's 1,000 times higher a spontaneous rate of mutation than other mammalian genes. And for that reason, it's sometimes also called somatic hypermutation, just to augment the mutation aspect of this. Now, at this point, you might be wondering, okay, just because they mutate, why does that make it better? And in fact, it likely doesn't. Usually, this mutation will just result in an unideal rearrangement of the genes, resulting then in a lower affinity to that particular antigen. So then the question remains, okay, how do the cells only choose B cells with the higher affinity antibodies? So let's talk about that. I think it's a really interesting process. As I said before, these follicular dendritic cells, or FDCs, don't express the class II antigen-presenting mechanisms, but instead express Fc receptors that can bind to antibodies, and then the complement receptors for complement activation. And they use these to present antigens bound to antibodies or complement proteins. So after reproducing in the dark zone, these B cells migrate from the dark zone to the light zone, where they need to get stimulated by antigens, otherwise they die. Antigen stimulation induces the expression of certain anti-apoptotic BCL2 proteins. So these B cells then, they take the antigen from these follicular dendritic cells, they use it, they endocytose the antigen, present those antigens to the follicular helper T cells, which also signal for survival via the CD40L ligand. So this process uses up some of the available antigen within the germinal centers. So after the end of round 1, those B cells that got fed the antigen go back to the dark zone, they reproduce, they proliferate, they rearrange and mutate their genes so they have different affinity receptors, and then they go back for round 2, except this time there's less antigen available. So only those cells with slightly higher affinities have a better chance of surviving. They eat up more antigen, they go back to the dark zones, they reproduce, they proliferate, they rearrange their genes, and they have antibodies with, again, different affinities, and they go back for round 3 and 4. And each time, more and more of the antigen is used up, it becomes harder to get, and only those B cells with the higher affinities end up surviving. So this is really just a process of natural selection. And for this reason, B cells with lower affinity receptors never survive this process. Now, during the final rounds of affinity maturation and the selection process, 2 cell types can exit out the germinal centers, one being plasma cells. Now, plasma cells are terminally differentiated effector B cells whose entire job is to make antibodies. And of these, there's 2 types. There's the short-lived plasma cells that are generated during T-independent responses and in the early stages of T-dependent responses that occur in the extrafollicular B cell foci. And there's a long-lived variety generated during the later stages of the T-dependent responses that we just finished talking about inside the germinal centers. So the precursors of these are called plasmablasts, which are typically found in circulation and do not express CD20, which is a marker of mature B cell zones. Plasmablasts eventually migrate into the bone marrow where they finish maturing and where most of their antibody production takes place. So to clarify some nomenclature, particularly in this chapter and onwards, whenever we say B cells, we're usually talking about undifferentiated cells that have yet to be activated. And then plasma cells are fully differentiated cells that are committed to producing antibodies. One big change in plasma cells from B cells is that they enlarge dramatically upon differentiation. This is in part because there's a large cytoplasm to nucleus ratio, the ER becomes more prominent, and this is all for the factory portion of the B cells. This is what's necessary to produce antibodies. And that in itself is also a big difference between B cells and plasma cells. B cells make surface-bound antibodies, so they don't really secrete antibodies, and then plasma cells produce the secreted antibodies. This picture on the left, I think, illustrates the difference between B cells and plasma cells very well. In addition to plasma cells, memory B cells are also generated in the germinal centers and are produced during T-dependent B cell activation processes. In comparison to plasma or naive B cells, memory cells have much longer lifespans for two reasons. One is that they have much higher levels of the anti-apoptotic protein BCL2, and furthermore, they don't require antigen stimulation in order to survive. Because they're made of the germinal centers, they typically have higher affinity antigen receptors compared to naive B cells. They've undergone isotope class switching, so they can express many types of Ig isotopes. And in comparison to plasma cells, they secrete no or very low levels of antibodies. So they themselves are not factories. They dedicate all their energy towards sustaining life and staying around for a long time until activated again, as opposed to plasma cells who spend an enormous amount of energy producing antibodies. Now, both traits of memory and high affinity maturation are highly desirable in the development of vaccines. Now, because both of these are dependent on T-dependent processes, the question remains, what do you do about antigens that don't stimulate T-dependent processes? What if they're from polysaccharides or from lipids? And this is where the concept of the hapten carrier effect and the conjugate vaccine comes into play. So in this case, scientists will take the characteristic antigen, be it a lipid or a polysaccharide, conjugate it to a foreign protein to form this conjugate vaccine. So from this, the B cell will recognize that lipid, polysaccharide, or hapten. The T cell will recognize and activate from the peptide or the protein. And then the T cell will further activate the B cell through the T-dependent activation processes. So these type of reactions have proven particularly effective in inducing protective immunity in infants or young children who are less able to make strong T-independent responses from polysaccharides compared to adults. So in short, during the initial antigen recognition process by the B cells, naive B cells become activated, and this could be both through T-independent and T-dependent, but in the short-lived follicular zones, which is a lot more rapid. Both of these processes generate short-lived plasma cells and lymphoid organs. They tend to generate a lot of IgM antibodies, to a much lesser degree IgG. The antibodies have a lower affinity, and these are induced by all immunogens. In the later stages, the response by B cells becomes much larger because T cells start becoming into play, right? So this is only with protein antigens, but the magnitude increases, type of antibodies produced change depending on the need, but we certainly start seeing a lot more IgGs, IgAs, IgDs, and Es for whatever specific purpose is necessary for that type of reaction. The antibody affinities are certainly a lot higher due to affinity maturation. And then lastly, in this process is where we get the memory B cells that can last for a really long time for subsequent reactions. Lastly, I want to touch on the ability of some secreted antibodies to inhibit continuing B cell activation by formation of antigen-antibody complexes that simultaneously bind to antigen receptors and inhibitory Fc gamma receptors on antigen-specific B cells. And this is a mechanism that's called antibody feedback. So we talk about a series of these Fc gamma receptors in the next chapter, but this is one that I'm going to introduce here. All these Fc gamma receptors bind to Fc portions of antibodies, as their name implies. So in this case, the Fc gamma R2b, And there's a whole series of these Fc gamma receptors. This Fc gamma R2b binds to the Fc portion of IgG antibodies, and this serves to inhibit downstream receptor signaling processes that halts activation and ceases antibody production. And we can see the effects of the deficiencies of these in knockout mice that are unable to generate these type of inhibitory mechanisms, and they just produce plasma cells that have just uncontrolled antibody production. And there's also some human autoimmune diseases like lupus that are linked to uncontrolled antibody production as well. So that's it for the B cell activation processes. In the next chapter, we're going to talk about the functions of these antibodies and the humoral arm of adaptive immunity in general. So thanks for listening.